Jordan Novotny here to discuss my Unit 2 case connections. We will look at two medical cases involving glucose uptake in a glioblastoma and a hypoglycemic infant. I do apologize in advance with this recording. You may hear my dogs clip-clopping around our hardwood floors and also maybe some firecrackers outside of our house as it is 4th of July. So I've used case 2 for my first case connection. The case reads as follows. A brain MRI shows a large glioblastoma tumor with areas of necrosis within the tumor. The tumor is the bright white area within the brain field on the MRI. A PET scan is also taken from the same individual following administration of fluorescently labeled D-glucose. Use the following series of questions to determine how and if glucose uptake differs in the tumor when compared to the surrounding tissues. In normal tissue, how is glucose uptake regulated in the following tissues? Liver, skeletal muscle, brain, and adipose. How does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the above image, and why would this be advantageous to the invasive tumor? What modifications may have occurred in the cancer cells to alter glucose uptake as predicted above? To answer the first question in this case, I will review normal glucose uptake regulation in 4 different tissue types. The liver, which is provided glucose by the GLUT2 glucose transporter. The skeletal muscle, provided with glucose by GLUT4. The brain, provided with glucose by GLUT3. And finally, the adipose, also provided with glucose by GLUT4, similar to the skeletal muscle. In the liver, the main regulation mechanism for glucose uptake is glycolysis. The liver primarily utilizes glucokinase, which is not allosterically inhibited by its catalytic phosphorylated product, glucose 6-phosphate, unlike its isoform hexokinase, which is found in skeletal muscle and other tissues. The lack of allosteric inhibition of glucokinase allows it to continuously take in glucose as fuel for glycolysis during periods of substantial glucose loading. There are 2 other important enzymes utilized within the liver during glycolysis. Phosphofructokinase 1, or PFK1, to convert fructose 6-bisphosphate into fructose 1,6-bisphosphate. This enzyme provides the irreversible step to commit the glucose to glycolysis and is allosterically activated by the presence of ADP or AMP to promote the production of more ATP. Inversely, the presence of ATP or citrate allosterically inhibits PFK1 so as not to provide an overabundance of energy. Liver-type pyruvate kinase, or LPK, is used to convert phosphoenol pyruvate into pyruvate within the liver. LPK is allosterically activated by the presence of fructose 1,6-bisphosphate, again provided by active PFK1. Similar to PFK1, allosteric inhibition takes place in the presence of ATP to prevent excess energy. Other metabolic pathways via the liver include glycogenesis, glycogenolysis, and gluconeogenesis. The chosen metabolic pathway is allosterically controlled. The skeletal muscle also primarily utilizes glycolysis for glucose uptake, mediated by insulin and facilitated by hexokinase activity. This is also true of glycolysis within the brain and adipose. Hexokinase has a much higher affinity and lower Km value for glucose than its isoform glucokinase. Glycolysis within the skeletal muscle produces lactate, which can be transported to the liver for gluconeogenesis. Now, how does glucose uptake in normal tissue compare to glucose uptake by tumor cells, and why would this be advantageous to the invasive tumor? What modifications may have occurred in cancer cells to alter glucose uptake as predicted earlier? Normal tissue tends to rely mostly on oxidative phosphorylation to generate energy. But it seems that glioblastoma tissue is able to shift the primary energy mechanism to glycolysis by overexpressing the GLUT1 and GLUT3 glucose transporters. This so-called hijacking effect allows for mass quantities of glucose to be rushed to the cancerous cytosol to be used for glycolysis, which is a less demanding energy pathway that does not require mitochondrial transport despite its low ATP yield. This increase in glucose uptake within the glioblastoma tumor explains why one is able to visualize the presence of the tumor on MRI and the PET scans following administration of the fluorescently labeled D-glucose. This mechanism has been coined as the Warburg effect, named after scientist Otto Warburg, who hypothesized this energy shift in the body's tissue following tumor growth. The benefits for overexpressing GLUT1 and GLUT3 transporters are very interesting. GLUT1 is traditionally used for transporting glucose in anaerobic conditions, which makes it ideal for transporting glucose to the intermediate zone of a glioblastoma, where the environment is hypoxic. And as a reminder, GLUT3 is the transporter used to provide glucose to the brain and is typically expressed within the neurons. Moving on to my second case, I decided to go with case 4, which reads as follows. A previously healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports she has not given the baby anything to eat. The diagnosis that is consistent with the patient's history and physical, presentation, and the medical team's findings is glycogen storage disease. Glycogen storage disease is better known as von Gurk disease. There are many different types of GSD, but this particular patient's history and physical reflect a classic type 1A presentation of von Gurk disease, which is a hereditary disorder manifesting due to a deficiency of the enzyme glucose 6-phosphatase. The role of glucose 6-phosphatase in liver glycogen degradation is the cleavage of glucose 6-phosphate into free glucose. If there is a deficiency of this enzyme, this will lead to a lack of glucose production, otherwise known as hypoglycemia, and lactic acidosis. Recurrent episodes of hypoglycemia and lactic acidosis may result in cerebral damage and abnormal cognitive development. The classic history and physical in patients with type 1A von Gurk disease include hypoglycemia and lactic acidosis, usually presenting from ages 3 to 6 months, with hepatomegaly and seizures. Hypoglycemic episodes appear while fasting between feeds. These presentations are consistent with our patient's age, her mother's report of jittery spells, the enlarged liver, and her inability to maintain consistent blood glucose levels while fasting. Finally, the administration of epinephrine is a telling diagnostic, as epinephrine is an important hormone in the mechanism of glycogen degradation within the liver. If epinephrine is administered, this should catalyze the degradation of glycogen to free glucose. However, it appears that epinephrine administration caused further lactic acidosis, likely indicating the diagnosis of type 1A von Gurk disease for the patient's medical team. Answer A, hereditary fructose intolerance, can be easily ruled out, as lactose from breast milk consists of glucose and galactose. And if you remember, the patient's diet has only consisted of her mother's breast milk. Answer B, galactosemia, can also be ruled out, as a newborn would likely present with its set of symptoms within just days following birth. Its common symptoms include feeding difficulties, lethargy, failure to gain weight, jaundice, liver damage, sepsis, and abnormal bleeding. These are quite different from the presentations found in our patient. Finally, answer D, glucose 6-phosphate dehydrogenase deficiency, is much less likely to be the correct diagnosis in this case for multiple reasons. This genetic disorder is highly uncommon in females, firstly. The defects are also associated with G6PD enzyme causes premature hemolysis or red blood cell breakdown. The common symptoms associated with this disease include paleness, jaundice, dark urine, fatigue, shortness of breath, and tachycardia. Again, not consistent with this case.